Hello, everyone. We're going to be starting here in just a minute. And in the meantime, thank you for your patience. All right, welcome everyone to today's session. Today we're going to be doing our session 1 of OmicsLogic Data Science. This is a program that is hosted by the Louisiana Biomedical Research Network. And so today we will have an overview of the program, but we will also start going into some practical aspects of how to load data in different formats, TXT, CSV, how to summarize that data. We'll try it both in R and in Python. But before we begin, I want to give a brief overview of what this program is going to be all about and kind of walk you through where we headed with this program and what are you expected to learn as a result of participating in this program. So again, today is our first session, and we're going to be speaking about different aspects of data science in application to biological or biomedical data, as those will be the examples that we will use. I will be one of the speakers, but together with me, we have Clinton Cunha, which will be doing part of the session. Hello, Clinton. But also we will rely on some materials that were prepared by our team, and you will be able to interact with them as well, getting help with all kinds of different technical issues that you might come across. So what is OmicsLogic all about? OmicsLogic is all about applying data science principles to different biomedical domains using omics data. And that includes oncology, infectious diseases, neuroscience, and agriculture. And so we call this program Data Science for Biomedical Discovery, or biology as a data science. It's kind of like the overall term for what we do. And what our company provides is we provide different tools and methods for big data analysis that includes processing, analysis, and integration of omics data like genomics and transcriptomics, phenotypic data like images, clinical data, and also we discuss this in the context of very specific project examples. We also provide a network of mentors, and these mentors come from different universities, including Louisiana State University, Georgetown University, and others. And our mentors are typically experts in different domains, so they come and they share their perspective on a particular project example. And we have a number of programs that are designed around that one-on-one or small group mentoring. We also provide access to training materials. So today we will take a look at some of those materials to get you started with data science. But when I mention data science, what does that actually mean? So data science is the convergence of methods to deal with data, store, organize, manipulate, ways to analyze that data or statistical analysis, and ways to apply that to a particular domain. So data science, typically when we hear data science, we think of it applied to many different domains: clinical, business, Internet of Things, finance, and marketing. Well, this specific program is going to be focused on the application to the biological domain. And so we will use research examples that illustrate how omics data could be useful in addressing some of the challenges in precision medicine and different aspects of biological research. So the program itself will cover some introductory material on statistical analysis and machine learning. We will discuss how that plays a role in experimental design. So how do we analyze data to spot real signal and differentiate from technical variation? We will talk about data mining and what it means to mine data for patterns and finding associations that might be meaningful, and how to also test those associations. And then we will talk about the complexity of biological systems and how that applies to the different types of data that are available and we will use as examples in this program. In the context, we will also try different types of tools. These will be big data processing tools using our Bioinfo platform, but also scripting in Python and R. And so what we hope is that as a result of this particular program, you will cover the following topics. So introduction to data science and bioinformatics, how the two are related, but also what are the specific aspects of data science that typically are not so covered as well in bioinformatics itself. We'll talk about data processing, cleaning, and exploration, the applications of data science and biomedicine. We'll use several examples for that. We will talk about different types of machine learning algorithms, and we will apply them to next generation sequencing data. We'll talk about the challenge of integrating various signals from X-omics or multi-omics data sets. And then we will also talk about the subject of deep learning and how that could be applied to omics, but also to clinical phenotypes. So as a result, what we hope to do is connect the research question or the real need that exists out there with different aspects of data analysis that has an impact on how we think about the data or the experimentation that generates that data. How do we explore that data and discover patterns that turn into hypotheses? How can we test those hypotheses, and what that can reveal about different types of biological experiments producing high throughput data that oftentimes does not necessarily fit our knowledge. And so how this data-driven approach to data analysis is critical for discovery, and how data science plays a major role in that process. So let me give you an example of why data science is such an important part, and how does it apply to such intuitive applications like precision medicine. So as we all know, precision medicine is the future of medicine, in the sense that we are relying more and more on complex data generated for clinicians to make decisions. And these decisions could be about treatment, it could be about outcomes, it could be risk stratification. And so in that process, we are also talking about data becoming more and more precise, or also complex. And so when we think of traditional medicine, which is medicine based on symptoms, those are at the top of the iceberg, or the clinical data that we can see and observe the manifestation of disease and symptoms that are clearly identifiable. And we want to connect them with the underlying molecular drivers of that process to be able to intervene. So to be able to spot specific patterns that can reveal some actionable insights. That is one example. Another example where machine learning and data science are very applicable is drug discovery and drug repurposing, where the length of time from ideas about what might work to actually getting a drug on the market is not only expensive, but also very long. And that's where data science and the application of machine learning will play a major role. So we'll talk about some examples in that, but also in clinical trials, how going from recruiting patients for clinical trials with a broad kind of high-level symptom-based recruitment is also changing, because now they're being recruited with biomarker, and that makes those very precise. And that makes that also very different from the traditional kind of landscape of patients and disease stratification. So we will also talk in this program about different types of omics data. So as you probably know, omics data could be classified into several different types. For example, phenomics talks about phenotypic data. Genomics talks about DNA variation. Epigenomics talks about epigenetic regulation and different signals that we can capture about chromatin structures and accessibility of DNA. We can also talk about the result of that regulation, which results in expression of mRNA and other types of RNAs that could be collectively called transcriptomics. And so when we talk about biomedical omics data, we talk about different examples with diagnostics for cancer biology, basic biology, where we try to understand single cell and tissue composition, or even looking at the impact of different variations at the microbiome, and for example, immunity to infection. Now, what characterizes all of these examples is the availability of unstructured data. So many times we rely on data that has been generated by processes which we can call high throughput. And what that means is that they are automated. And because of that automation, a lot of the data is generated in unstructured formats, which, for example, could mean the collection of short reads that contain sequences, abundances, and the process of taking that unstructured data and turning it into something useful is what we call processing. So for example, we can process whole exome sequencing and whole genome sequencing to identify the actual mapped reads to a reference genome. And then we can ask the question, well, where are the changes that are significant? We can talk about DNA methylation and chip sequencing, where we take a look at immunoprecipitation and methylation processes that help us extract meaningful insights about epigenomic regulation or epigenetic regulation, sorry. We can look at RNA sequencing or levels of gene expression, and we can also look at peak calling, for example, for proteomics and metabolomics, trying to understand what kinds of proteins and metabolites are being expressed in the sample. So taking that raw data from images or reads and turning it into structured data is basically converting that raw data into a table. And once that table is there, we need to understand the different aspects of that data, which might include numeric data, continuous, but also categorical, where we have names of samples, groups of samples, names of genes or proteins or whatever else it might be. And so in the context of data science for biomedical discovery, we will talk about how we can do data processing, how we can explore that data, and how we can perform data analysis relying on what we understood about the data sets that we have for some kind of inference. So for interpretation, really, how can we take what we have found in a given sample and generalize it about the general population, disease, patient, etc. So let's take a look at an example. First of all, genomics, transcriptomics, metagenomics, and epigenomics. All of these types of data sets are complex. They involve many different features like genes, metabolites, proteins, and typically as a data scientist, our challenge is to convert it into something that is insightful. So that might mean some kind of transformation or some kind of organization of data, plus some visual tools to help make that interaction with data intuitive for people that don't necessarily go through the process of making those steps, but actually can take a look and it's clear what is the message, what is the status. And so we'll learn in this program how to perform these steps, the transformation steps, how we can visualize appropriate types of data with appropriate types of visualization, and how we can use advanced techniques like principal component analysis or hierarchical clustering, like in these examples, so that we can extract some meaningful information and share it with others. We will also take a look at the relationship between what the data means and how it applies to the biological context, like taxonomy, and how we can use our understanding of the data properties to, again, communicate the differences in our data, in our biological samples, with some clear visual messages. So to talk about that, we need to understand that data comes from many different sources. And whatever that source is, when we make it into a structured data set, we have to typically go through a series of steps so that we can prepare that data for analysis. And so that will be the first part of our program. We will talk about several different examples of working with data sets like that. Then for us to work with a computer, we need to convert the full table or whatever structured data we have into specific objects. These could be vectors, matrices, arrays, or data frames. And so we'll talk about how these different types of objects are useful for storing and operations with the data so that we can extract some associations, correlations, visual information, etc. We will also talk about how visualization of this data is also guided by different types of data properties. So we'll talk about what is categorical data, what is continuous data, how do we combine them together to summarize data. For example, if we have metadata about different objects in our table, how do we create what's called crosstabs or summary tables that essentially give us a summary of what is in the data. We'll then talk about some useful statistical measurements or properties of data that allow us to spot major patterns right away. So you might think of it as box plots, for example, that summarize some elements of distribution, mean and interquartile range, so that we can clearly see major patterns in large data sets. And we'll also talk about scatter plots and different ways to prepare data for scatter plots that are informative so that we can spot relationships between elements in our data. Now, importantly, because we're talking about the biomedical domain, we also will have to touch on data normalization and understanding of how the data was prepared. So a lot of the properties in the data might not fit the assumptions of the methods for exploratory and in-depth analysis. And so we'll talk about steps to normalize and change properties of data without really changing the data itself, so that we can test things like technical replicates or biological replicates, and how useful are they in being used for downstream analysis. Then finally, we'll talk about exploratory data analysis and what that really means. How do we use elements of machine learning and statistical testing to be able to understand how the patterns characterize the data set in general, and what does that mean for downstream statistical or comparative analysis? When we talk about statistical analysis, we'll talk about how the analysis itself of a subset of data is intended to really tell us something about the greater population, and how different elements of the statistical analysis, like p-values, false discovery rate, and other log-fold change and other measurements, can help us find a statistically significant signal that could be used for predictive analysis or for inference. How can we use those different statistical measurements to really give us reliable results? We'll also then talk about data mining and classification. So we'll talk about different methods for data mining, like principal component analysis and different types of clustering, and how to then also rely on different elements of classification, like decision trees or random forests, to analyze and classify our data, but also to do feature selection. So finally, we will touch a little bit on different algorithms behind some of these methods, so that we understand what goes into the different steps of an algorithm that can help us understand and extract meaningful features inside a data set, and how we can use the different parameters included in that algorithm so that we can optimize it for the type of an objective that we have, like feature selection using stepwise linear discriminant analysis as displayed here in this slide. Finally, we will touch on elements of deep learning. And so, as exciting as it is, there are also many different challenges with deep learning that are specific to different types of data, different types of network architecture, and different types of applications where they have been demonstrated to work better than, for example, classical machine learning, and where they are performing in such a way that they're addressing other challenges that are not related with, for example, the optimization and the accuracy, but are related to the type of data and the challenge with really preparing data for analysis and for training. Now, all of these are some of the conceptual topics that we will cover, but we will also go into some practical coding sessions. And in those coding sessions, we will take a look at how to implement some of the analysis that we described, and find useful pieces of code that you can save for your own personal repository of coding libraries that you will rely on to work on an independent project. And so we hope that after this program, many of you will be able to benefit from some of these coding exercises and actually take it further and apply them to a data set of your own. So here are some coding challenges that we will go through, including some of them today. We will learn how to load data from different formats. We will learn about summarizing, preparing them with different types of transformation techniques, how to perform statistical analysis, and also how to perform different types of visualizations and how they are useful for different kinds of questions. As I mentioned, this will also include R and Rstudio, as well as Python and some development environment like Visual Code Studio. So we will talk about how we go from just exercises to then working in an environment that you can set up, how to load libraries, how to organize your workspace, how to save different files in such a way that you can find them, and then also how to run your analysis and save results that you can use or maybe publish or maybe present to somebody else. Now, we hope that in that context, you will also get exposed to some of the useful coding practices that are typically used for reproducible research, but also for teamwork. So oftentimes you will have to share your code with somebody else and they will have to navigate what is in the code, what was done. So we'll talk about how to use comments, how to organize your code, how to run functions and how to call the functions with different parameters that you need to have that function work. We'll talk about how to troubleshoot your code and kind of understand what the issues mean and where do they come from, how to find answers. We'll also talk about sharing your analysis in Jupyter notebooks on GitHub and Google Colab, and we'll take some examples so that we can also prepare some visual interactive visualizations using Plotly. Now, in the context of the program, we will go through several example data sets, and all of these data sets come from our reviewed set of projects. These projects have been reviewed for some conceptual approaches, which relate to different types of machine learning methods or different molecular biology or precision medicine topics. So we will really take a look at how the biological or the application side connects with the types of methods that were used and for which purpose. We will also understand how the algorithmic part or how the analysis part fits in to some of the challenges that are being presented and how do these algorithms actually address the problem by integration, classification, and predictive analysis. Now, we do have a lot of different project examples already. You'll be able to find all of these project examples on our portal. And so this brings me to how this program is actually going to work. The way it will work is we will have interactive sessions like today. I will pause in a second and we'll actually interact. So I'll ask you questions and ask you to do certain things. We will also rely on some self-guided study materials. These study materials are organized in specific lessons and courses that will be useful for this program, but also they will include some practical assignments. And so in a moment, I will guide you through some of the resources that you need to have access to benefit from this program the most. How many of you already have an account on learn-omicslogic.com? So please put number 1 in your chat if you do have an account. If you do not have an account, okay, thank you, then please go to learn-omicslogic.com. Okay, I see that a lot of you already have an account. So let's go to this account together. Okay, if you do not have an account, this is what we need to do next. So this is the portal. As I mentioned, learn-omicslogic.com. I'll give a moment to everyone who's just joining us for the first time to create an account. How do you create an account? You will find on top of this menu a button to log in. And so once you are here, you can create an account. Okay, so I will wait a minute for those of you that do not have an account yet to create an account. And once you do, please log in. For the others that are already registered, please sign in and go to your profile. Okay, I see some people are signing in. Okay, excellent. So once you are in, you should be able to see your email address right here on top. And when you click on this, you have a user ID. Now, because this program is supported by the Louisiana Biomedical Research Network, or LBRN, for those of you that are here and are a part of the program, I ask that you copy this URL and put it right here in the chat box, okay? So that way we can have a registry of everyone who is participating in today's session and who is either joining the program already or is planning to join, because today is our first session. So please share the link to your profile. If you don't want others to see it, you can do it directly to me. Okay, thank you, Navneet. All right, excellent. Thank you, everyone. So I'll just, again, give just a couple of minutes for everyone to share that link. Thank you, Madeline. And Rebecca, Ayomide, Dua, Navneet, Ethan. Okay, excellent. Thank you, everyone. Now, especially for those of you that are part of the Louisiana Biomedical Research Network, we do need for you to fill in, you need to create a profile. You don't have to use your personal image. You can use any image that you want. You can also put in a little bit of a bio right here by clicking on Edit Profile. And I will explain why this is going to be useful for those of you that are a part of the summer research program with the Louisiana Biomedical Research Network. All right, thank you, Hidayah, Durba, Amrit. All right, great. So as you go through this program, you will see that all of your activity is being logged. And this is to help you see your progress and understand where you are compared to where this program is taking you. The objective of this program is to get you to 10,000 points. So you can see that I just crossed over 10,000 points not so long ago. So whenever you get to 10,000 points is when you can start working on an independent project, which is really the objective of this program as well. It's not only just to learn, but also to be able to apply what you learn to a data set that you find and create either a project proposal or work on a complete project on your own. Here you will find the different coursework that is available and that you are working through. So that means that you are completing the projects, like I completed this project and this course, and I can see my certificate. And some of them I just started, so you can see that they're at 0%, some of them I am working on. Also, for those of you that have already registered, and I noticed a few of you from the LBRN did not yet have an account. Now it's important because now that I have your account information, I will be able to add you to the program. So the program that we are in is LBRN OmicsLogic Data Science. So if you go to this program link, I will share the link in our chat. If you have already completed your registration to this program, this is what you should be able to see. You should be able to see your profile, see here the overview of the program, which is right here, see the coursework included in this program, and these are the courses that we intend to go through. You will also see that you can book a meeting to discuss with us any challenges. You will have access to our Slack channel where you can ask questions from our support team and also get to know the other participants of the program. And finally, once you complete 10 sessions of this program, you will receive a certificate about your participation. How do you know that you've completed a session? So down at the bottom, underneath this overview, you will find the program schedule. In the program schedule, you will find a Zoom link, so you know how to join today's session, for example. And you also have this button that says complete session. So once you press on complete session, it says session completed. Down here, you can see that now I have 1 out of 10 sessions complete. So every time that we have a session like today, you complete them, and that means that you can work towards your certificate. Now, in addition to this, as I mentioned, we will have some asynchronous materials for you to work through that include some active learning which will include some coding exercises and practical exercises, including quizzes. So that will help you understand what you have learned, what you still don't know, and where do you need to focus for you to really master data science for biomedical discovery. So what are these courses down below? These are some of the coursework that we will plan to complete during this program. First of all, I would like to ask all of you to go to Introduction to Bioinformatics. This is an important course, not just because I'm assuming that all of you do not know about bioinformatics. I'm sure that most of you already know something about bioinformatics. However, this course also allows us to get to know about you. And so in the first lesson, you will find here a survey on your background, educational background, including your bioinformatics experience and what you are interested in. So that will help us adapt our materials more to your needs. So let me pause here for a second. Does anyone have any questions so far? If you do have any questions, feel free to post them in the chat. Okay. So no questions so far. So I'm assuming that everything is very clear. So the first thing that we talked about is that we have this online repository of asynchronous resources on learn.omicslogic.com. Those will include some coursework, but also, thank you, Dr. Candace, as well. That includes some coursework and some practical project examples that you can start seeing how to apply some of these methods to real challenges. Also, we talked about our personal profile. On the personal profile, if you would be able to complete your image for your profile, write a little bit about yourself. You will see here points accumulating, and you importantly will find here a subscription. So for those of you that are joining the program, you will need a starter subscription, which is different from the basic. That means that you have access to premium courses, and you will have a badge. That badge will be from your program. So this program is called LBRN Data Science. If you don't have those, what I will do today after the session is go through the profiles that you have submitted. And if you are in the list from LBRN, we will assign you to that program, so you will gain access to all of the resources. If you do not have it and you're not a part of LBRN and you're waiting to join, please proceed to the program page. You will find their directions on registration. Navneet is asking, will I share these recorded videos as well? So we have our program page. Let me just show you the program page again. So if I go to my profile and I'm registered for this program, I can go to the program page, and then here, after this session is over and the recording is posted, you will find the recording right here. And then the next session will become a Zoom link. So everyone who's a part of this program will have access to all of the recorded sessions, and we will also guide you through some asynchronous materials that you can complete. Okay. All right, so hopefully that's enough about the program. If you do have any other questions, post your questions in the chat box, or if you have those questions after the session, please use Slack. We have a dedicated Slack channel for this. You can find the Slack channel by going all the way to the bottom and click on Ask a Question. It will redirect you to Slack. So on Slack, we have a dedicated channel, again, for the program participants, and you will be able to ask questions. For example, I posted today the Zoom meeting link right here. All right, so if there are no other questions, I would like to introduce you to Clinton Cunha. Clinton has a master's degree in molecular medicine as well as in bioinformatics, and is experienced in different aspects of data science, including Linux programming, R, and Python. And so I will turn it over to you, Clinton, to take it from here. Thank you.
Thank you, Ilya. So today we're going to go over some introductory Python and R that will help you load different data sets into your own programs. So could I get just a rough idea if most of you have an IDE installed on your computer? So maybe put a 1 if you have either R or Visual Studio Code installed on your computer. Okay. Nice, nice. If not, it's fine. We can also go over. Nice. Okay. If not, it's fine. We will also go over how to install that through the tutorial. So let's start off with R programming. Okay. So if everyone could go to their course page. So to go to this course page, you'd have to go to this link. I'll paste it in the chat. And then we have to scroll through the courses. You can also type it. And let's scroll to R Coding 1, Getting Started with Bioinformatics. Okay. So if you can click this link and then click the first lesson, Loading Data in R. Okay, I'll give everyone just a second to catch up. Perfect. So there is a tutorial on how to download R and Rstudio. We can go over it near the end of the session, but most of the activities that we'll do today will be on the website itself. But if you don't have R Rstudio installed or an IDE similar for Python, please reach out to us and we can also help you step by step with how to install it. Okay, so I prefer Visual Studio Code just because I find it more user friendly compared to PyCharm and other IDEs, but everyone has their own preference. I would recommend starting off with Visual Studio Code for Python just because it's very user friendly. Okay. So hopefully, so you can take a moment to read through this. So this is essentially giving you an introduction of R, and R is not only limited to biomedical data, but other facets of the industry also use R for statistical analysis. So it's a very handy programming language to have because it's a data science tool. Okay. So after you went through the a little description of what R is, we can jump to how to put in files within R. So maybe you have different types of data files, whether it's TXT, CSV files. Most of those are the common formats that we use. And you can specify different coding blocks or coding statements to read that particular file. So over here, if you want to import and view a specific file into R, use a function known as read table. And it does have some extra parameters here. This tells you how you want the columns to be separated. And header equals true tells you if the first row means the headers of those particular columns. So depending on the type of data set you have, you might want to put header equals false or header equals true. So you can take a few, I'll give everyone around 2-3 minutes just to read over this, and then we'll do the practical coding exercises right below, okay? So take a moment just to read this material and get a sense of what's going on. And if you have questions, please write it in the chat, specific questions as you read along. So I'll give you around 2 minutes, so 2-3 minutes to read this over. Yeah, that's true. So sometimes, sometimes, yeah, code sounds better in our head sometimes, but while we do it, there seems to be like different issues that arise. So I can completely relate to that example. And depending on your coding experience, there's a very uh, oh, so that's that's a common thing with programmers in the sense that, you know, sometimes they have an idea in their head, but they need, like, they might Google things or go on Stack Overflow, different websites to piece it together. So you, I think that, like, some people joke around with programmers by calling them great Googlers. So it's, if you know the fundamental pieces and figure out how to put them together, then you can build on those small steps for bigger projects. So, you know, if you do feel nervous, don't worry. This isn't meant to be a super intensive and difficult course. It gradually builds up, and there's an asynchronous part to it. So it starts off with simpler exercises and then builds up to more advanced topics. So yeah, every programmer starts with or data scientist slash analyst slash programmer starts with a more simpler method of coding. Okay. Oh, okay. So if you do not see R, let me just go back to that portion. I'm getting some messages that some people can't see this particular module. So back to the courses page. If you scroll down, go to number 2, and you should see an option saying getting started with bioinformatics R coding. Hopefully the person who could not see this can maybe clarify whether they can see it. Okay. So here is the R coding part, R coding 1, getting started with bioinformatics. And then all you have to do, perfect, awesome, great, no problem. Okay, then you click that. And then you click loading data in R over here. So you click that, the first module. And then you should see the different parts to it. So there are 4 parts, and I gave everyone around 2 minutes just to read this aspect. So we can try it. We can try it now. Okay. So let's go to the next part. Try it yourself. So over here, essentially what you're doing is you have a data set over here. We put it up for you, and you have to copy that data set and read it into your own into this online IDE, and then it will print that data set for you. So over here, I right-click here, and I click copy link address, and then I should put that link address into this code right here. So I click paste. Make sure you keep the quotes. Okay, so the quotes should be there. And then you should run that particular snippet of, okay? So what we're doing here is we're reading this text file directly into R. Okay, and we're separating each column by the tabs that's separate each column. So we specify this /T, and the header is the first row of that file. So if you click that file here, you can see the first row is the header of that data set. And then each of the columns are separated by huge tabs. So if there were commas between these columns, you'd have to put comma instead of tab. Okay. Can someone answer the question? If there were periods between the columns, what might you have to put here? If there weren't tabs, there were just periods, what would you make as the separator? Yeah, perfect. Yes. So right now the separator is tabs because, as you can see, there are large tabs between each of the different columns. So there's one piece of data here and one piece of data. But instead, if there weren't tabs and there were other types of separators like periods or commas, then we might want to change that into the relevant separator. Okay. So perfect. So for commas, it depends on your data set. So a lot of people, depending on their research methodology, they might not keep their data clean. So this is a very great example of clean data, but some data, they might not spell specimen correctly for all of the specimen words, or they might not have equal tabs between everything, and there might be a period here but no period there. So you'd use a comma if there's a clear separation between the columns with a comma, but over here there's a tab that separates the columns. So in this case you would use a tab, but in other cases you'd use a comma or a period. What's the default? That's a great question.
Let me jump in here real quick. So as you can see in the future code blocks below, there are different options to read. You can read CSV, which is specifically for comma. You can read table, where you need to specify. You can read XLSX, which you need a special package for. So these are really different examples. But in our case, what's important is that you first identify what is the type of file I have, and then based on what the type of data you have, you choose which option you need to use.
Yeah, that's a great explanation. Yep. So yeah, depending on the type of file you have. So over here we use XLS. Over here we use CSV, and you copy. Please take a moment to copy these links and paste them into the path name, so you can see how this exactly works, and click run. So over here, we're using a library to help us, read.xls. This function reads XLS files directly, and then we get our data set printed on the console. Okay. Take a few moments to go through this. And if you do have R installed, you can even copy that relevant code and run it on your Rstudio. So over here, you can put the same, since it's a URL, that also works. And you can read the table, or you can download the data set onto your laptop or computer and change the path name. So if this is the exact same thing on the website, but on my local machine. So you can also do something similar. We can go through the steps of downloading R and Rstudio near the end of the lesson. But for now, just focus on the online content and try to make those examples work. And let me know if you have any questions, if you're stuck trying to run some of these code snippets. And here are some extra files that you can run. So here's, this is a nice enlarged data set. So this is a breast cancer data set. And it has different labels and error classes, and along with their gene numbers. And you can read these files into your code here in a similar way. So paste that. CSV, read.csv, that should work. Great. Oh yeah, because this one, the answer is incorrect because it's expecting the data set up here, but it's still reading the file. Okay. And there's another. Oh, okay. So if anyone, just to reiterate, what we're trying to do here is we're trying to read different types of files into R. Okay. Instead of downloading the R programming language onto your computer right now, we're reading these files onto our online R code console. Okay. So you, instead of having this on your current screen, we're running the exact same thing onto the online console. So you don't have to install R right now, but we will go through the steps after near the end of the lesson, after going through some of these online coding exercises. So I didn't mention that everyone should take a few moments to read this snippet to get a sense of what we're doing. And essentially, to summarize what this is saying, is we are learning how to read different file formats, such as Excel files, text files, CSV files, into R using a variety of commands. So some of the commands include read.table for TXT files, XLSX for Excel files, CSV, read.csv for CSV files. So these are 3 different commands to read 3 different file types. So depending on, because this is a data science course, you will probably encounter these file types when you're analyzing different types of data. So you do need to understand at least the code syntax and how to do this for your own future data sets. I hope that clarifies the concept. And if there's someone that did message me, and I hope that clarifies it, if you could message me back indicating if that gave you some clarity, that would be appreciated. Okay. So these are just extra TXT, CSV, and Excel files to try to run over here. And you can copy the same code and run it in your own Rstudio if you have it. So here, I use the direct link to Code Omics, but you can also download it into your own system and copy that particular path name. So like over here, I have these test files, and then you open up your properties and you copy your path and you paste your path over here. However, the brackets are different. So instead of these brackets, you have to use backslashes. Okay, so the way R and also Python reads path names is that they read it through backslashes. So I won't go into this too much because some of you might not have R installed. So I'm going to skip this for now, but we're going to talk about how to install R later on. Okay. So hopefully, could I get a indication? Could you write a 1 if most of you have tried these exercises online and hopefully they're working? Could I get a 1? Perfect, perfect. Okay. What? Okay. We'll okay. Let me help you for one example, and hopefully, not sure where to try. Oh, okay. Okay. Maybe you. So to try this particular example, click number 2 down here. So there you have to be on this website. So I'll copy this. Okay. And then you have to click page number 2. Okay. And then you need to click R coding 1, getting started with bioinformatics. So you have to click that. And then then you click loading data in R. Perfect. Great. Great. And for Rebecca, for the first exercise over here, please click this button here. So you have a few options down here. So you can copy your code with this. You can reset your code. You can clear the console with this. So please right click the here over here so you can get open link to new tab, open link in new window, and click copy link address. Okay. When you click copy link address, you're going to copy the path for the location of the here file. Okay. And once you copy the path, take a moment to highlight the path that's in the console over here or in the script over here. And then right click and paste that path. So do you have something like this on your screen? Yes. Okay, perfect. Yes, perfect. And then click run. Did you see a lot of confetti fly all over the screen? Yeah. Yeah. Okay, perfect. Awesome. Great. Yeah, that's well, yeah. We don't have. Oh, okay. That's interesting. Because yeah, if you click run, you should say completed and the answer is correct. And you should have some confetti flying in the screen. Maybe it might be a browser specific thing, but it's fine. That's just an extra fun component of the code. Okay. So great. So most of you did this, and you can click complete and put some feedback on the complexity of this. This is meant to be easy to get you into the coding like habit and to get the data set you're interested in. So now after doing that, let's jump to Python. Okay. I personally, I like Python more than R, but that's just a personal opinion. I feel like it's just, there's more versatility, but that's just me. Okay. So you can go to, I believe it's in 3. No, wait. No, it's in 1, sorry. So go to 1 and click getting started with bioinformatics in Python. So this is the course that we're going to do now. So click this. Okay. And then click loading data right here. Okay. Okay. So these are some instructions to download Python on your own system. Just like the R course had some instructions, there's also a Python equivalent. So you can take some time to install this on your system, but preferably we'll do it later on or because of the time constraints of the session. But we will help you with it if you do have issues. Okay. So that the first part is about installing your IDE and Python. So you have to do both. And then please take around 2 to 3 minutes just to read the learn the code portion, because this is important. Some of this, it's good to read through what's going on to get a clear idea, and then we'll implement it in real time. So I'll give you 3 minutes. So please take 3 minutes to read this. Okay, so could someone type the extensions of the files that we're going to load in with Python? What are the names of the extensions that we're talking about for today's session? You can type the 3 side by side in the chat. Great, great. Yeah, perfect, perfect. Just like the R module, we're going to learn how to load XLS files, CSV files, and text files. So these are some common file formats that are available for different that data sets are available in. Perfect. So there's one key difference here between the way the Python module works and the way the R module works. So the R module is more memory intensive. So our auto grader takes your code input and sends it to a Google Cloud server and compiles the code over there and returns the output back to the online coding system. However, Python's less memory intensive and it can be handled within your Google Chrome browser. Therefore, we can directly install some packages over here and we can actively run the code on this browser. So that's one key difference to the way we'll put in files within this module. So let's see what that means in practice. So if everyone can take a moment to import Pandas and Numpy. So these are 2 data science packages in Python that are useful for data curation. So maybe please take a moment to click run and this might take a little bit of time to import these packages onto your system. Okay. And one key thing about this module is that I can't, okay. For some reason, I cannot annotate, but we are going to load the file down here into the drop file over here. So the code runs from top to bottom. So before you run this code command, you need to run this code, or you need to download this text file and drop it over here, and also specify the name of that text file in the code block. So I'll show you what that means in practice. So let's say I have this file over here. So I click it over here and let me save it somewhere. So let me save as. I'm going to save it here. I have a folder here, test files. It's already there. You want to replace it? Yes, I'm going to replace it. Okay. So I'm going to show this in my folder. My folder is right here. Okay. So I have this data set right here. So if I go back to this portion, I open this test files folder and I drag this text file into drop a file. You see how this changes color? So you bring it over here until it changes color to just orange, and then you drop the file inside. And then you should notice that it dropped because you see the name of the file inside this box. Okay. So then you can copy the name of this file and then you can put it in this location. So I'm going to reset this code just so it, okay, I guess this was already reset. And then if it looks the same, just paste that particular file name within the quotes and then run this script. So right now you're importing and viewing a text file, and then you should see the names of the columns and the values on the console. Okay. So hopefully everyone's, can everyone do that? Can everyone put a 1 in the chat just to see if that's working? Because the rest of the exercises depend on 1. Okay, it seems like we're still doing it, so that's fine. Okay. So essentially you download the file with the here link over here, and then you open the folder location of that file, and then you drag that file into the drop a file portion, and then you should see the file name pop up to the right over here, and then you copy that file name and you put it in the single quotes, and then you should click run. So then your code will run. Okay. Perfect. So in Python, they, instead of them saying data frame, they use PD, and it's in reference to this package. So this package helps you visualize data frames in Python. So a data frame is just a fancy way. It's just a fancy word for the way this data looks. This is a data frame. So it, with columns and rows. So that's what essentially is happening in Python, but it doesn't look as graphically appealing as this on R, but it's the same thing. Okay. And the same sort of parameters apply with over here, the tab and the header equals 0 portion. Okay. And please take a moment to try the other 2 exercises down here. So this is for a CSV file, and in this CSV file, you have to do the same process. So you click here, you download your file, your data set. You have it in your downloads folder. Sorry. So you have your downloads folder right here. Oops, there goes my. Okay, so you have your downloads folder right here, and then you copy. You can drag it directly from downloads into the next portion up here, and then you should have the file name here. Then you copy it, and then paste it down here. Okay. And then you should click run, and then it should work. Okay. So take a moment to go through this. Try. Okay. Yeah, so this is done. Great. And then we'll go over this data types portion. So here you can click this file, download the data frame data types. So there are different data types in Python. So it could be numerical or text, and amongst others. We'll see it over here. If you click the location of this file in your downloads folder, please drag it back to the drop a file. So this drop a file is for this next portion. And then click run. And then it should you should see different types of data types over here. So you have float values, and it says, this one it should be text to my knowledge, but let me double check. I believe it's text. One second. Sorry. Yeah, so the data frame one is, the float means the numbers on the right. the 23 point, the ones with the decimal, but O should specify the specimen name. Okay, so if you have data types with text and numbers, Pandas uses the O operator. So if I open that data set that I just downloaded, right here. So yeah, so the first column has text and numbers. So that's why it's an O data type. So it has a mix of both. So that's why Numpy detected it, or Pandas detected it as a O data type, which is a mix of both. Where do you click download? Oh, so here, if you click here, if you click this button, the file should pop up in your downloads folder. But that's how it is for most computers, but maybe in your computer you set it to a different one. So if I click here, I have the file downloaded on the bottom. And then if I click show in folder, it's in my downloads folder. So my downloads folder is here. It says users, me, and the downloads. So it would be right here. Okay. Perfect. Take a moment to try some of these extra data sets. So you can try the text file, the CSV file. And this function essentially just it prints the topmost rows for your data set. So if I were to run this portion, so let's say, no, I didn't put my file in, sorry. So let's, here's my 15 genes. Oh, sorry about that. I'm not sure what happened. I think too much data on my computer. Sorry about that. So yeah, when I download this file, and I drag it up here, and I copy this here, and I put that over here, and I run it. Okay, so it's when you have errors, it's good to sometimes clear your console and rerun the portion so you can see it. So the head. Okay, so don't worry, we're almost done. I just wanted to finish up with this coding exercise. So this is the head portion, and then this is to read this format, and this is just to show you the first 5 rows of the 15 CSV files, or the 15 rows of the CSV file. So great. So this is the practical coding session for today, but we'll do harder and more complex things later. I'll now pass it on to Ilya to talk more, to finish off today's session.
All right, thank you very much, Clinton. And hopefully everybody found this useful to understand how to do some of the basics. Let me briefly explain what is coming up next. Where is this program going? So the first part of the program is going to be introductory. Introductory means that today we covered omics data types and properties, and we briefly discussed how to load them. Now we will talk about big data challenges and opportunities. As you've seen, these are very small data sets. So then we will talk about where do we find real data, how big is it, and what are some of the challenges associated with extracting meaningful information from them, as well as what are some of the opportunities with so many data repositories growing out there. How is this going to be useful for you? Then we'll talk about cleaning, loading, and processing data. We'll talk about exploratory data analysis. How do we explore what are the different patterns, and how do we find interesting patterns that are signal and not noise? Then we will talk about how, practically speaking, in your own environment, do you set this environment so that you can handle large and complex data? And so that will mean, how do you combine access to cloud computing, some of the requirements for your own personal system, where do you store files, how do you store them, how do you organize them, and how to start loading all of these libraries into your own system so you can run it. And then also, where is this output going to be? So how do you organize the outputs from your pipelines, and how do you actually make sense of the steps that you need to be taking from the beginning to the end? In the second part of this program, we will start talking about machine learning and artificial intelligence. So machine learning in general is divided up into unsupervised and supervised machine learning. And there are many different methods out there that are typically used for analysis of large and complex data sets. And what we need to understand is, first of all, what is the logic behind what we want to do and what the method can do. And then how do we also combine some of these different machine learning methods to get us what we actually need. We then will talk about how, in supervised analysis or classification, we can train a model based on a data set that we've prepared, and then test the understanding of the model of what is in the data with some validation. So we'll talk about model accuracy, how do we measure it, ROC curves and area under curve, and then how do we compare different models in terms of what they're able to give us. And then finally, we'll talk about how to actually get results with machine learning. So machine learning is not analysis, a sense that I get data, I analyze it, I have results. Machine learning is best used when we have continuous data that we have to deal with. And so there's a whole process of setting up pipelines for machine learning and monitoring them. So that is something that we will cover. And in our next session, as you can see here, we will talk about big data challenges and opportunities to kind of take us past this initial step. Now, for those of you that joined today and do not have the Rstudio or Visual Code, this would be the important step before we meet next time. And also, I would highly recommend finishing Introduction to Bioinformatics and some of these practical initial steps. Now, if you didn't register yet, that is fine. You will see that most of this session's course is free, so you can kind of participate in everything that we do. If you do want to join our next session, you have to complete your registration before then. And please have your Visual Studio and your Rstudio installed. If you face any challenges, do let us know before we meet. So you can email us or you can let us know. Kisembo, I will definitely talk to our marketing team to send you the PayPal option. We can try that as well. But unless anyone else has any other specific questions, okay, thank you, Clinton. Homework for next time, complete Introduction to R and Introduction to Python. So Introduction to Bioinformatics in R, Introduction to Bioinformatics in Python, and also the course Introduction to Bioinformatics. Now we have another 10 minutes left for those that do not have Visual Studio Code in Python or Rstudio installed. So if you do have challenges with that, maybe you can stay and we can walk you through or answer any questions about that. If you don't have any questions, everything was clear today, you're ready, please complete your profile, start working on that coursework, and we will see you this Friday. Clinton, I will leave it up to you if anyone has any issues installing those, right?
Okay, yeah. I'm ready for it.
All right, so the only thing that we need to know is who has issues,
Yeah.
and then we can help them. All right, well, thank you, everyone. We'll see you next time.
So, okay. Yeah. So you're welcome to leave if you don't have issues, but if you have programming and other course related issues, please stay and we can go over them or try to get through them. Thank you for attending, everybody. See you guys soon. Okay. So I'm assuming everyone here either has an issue downloading RStudio or R, or Python and Visual Studio Code, or PyCharm, or some other IDE. So could you put a 1 if you have an issue with that? It's yeah. This is meant to be the first intro course, so don't feel shy if you, yeah. Perfect. Okay. Let's start off with RStudio, and then we'll jump to Python. Okay. Okay. So good old Google is my friend. So first thing we need to do is install the R programming language. So R programming language install. So this is independent of this is independent of RStudio. So if everyone can, I'll paste the link in the chat as well. So if you're, oh, okay. Any Mac users? I don't think it should be an issue, but if you're a Mac user, okay, great. Let's try to figure that out. 1 second. Some Mac. Yeah, I'm a Windows user myself, so we're going to have to. Yes, okay. This is R for Mac OS. So you'd want to choose. You'd want to choose the first one. This one. Okay. This is directly from the R website. So this won't work for me because I'm a Windows user. So we're going to have to figure that out together. Okay. See, this is a good one. Okay, let's see. Does Mac installing R, goes to this website. I believe it's the same website we went to. And yeah, it's the same link, but this is an older version of R. So you have to click the link I sent you. You still have to click this. And then there are a couple of installation instructions that you have to follow in Mac. So if you want, you can share your screen. We can do that one at a time. So yeah, that's an option. Okay. So first download this file. And then after you've downloaded it, you should be able to click on that file and it should download into your computer. So it should be like a .exe on Windows, and it should automatically guide you through the steps and accept the default options. So we're not, the default options are fine for our purpose. And most people install the default options. So. So let me know your progress to the Mac users installing R, because just let me know what's going on. Whether it's like you can write a little statement, or you can even talk, like use your audio and tell me what's going on. Okay. Perfect. Great. Okay. Now you have R on your system. Okay. Now you need Rstudio. So Rstudio it's the IDE for R. So Rstudio download Mac. So, oh, okay, you're downloading. Perfect. So Rstudio, and you want to choose the free version. Okay, this is for Windows. Windows 10 right here. So this is the operating system that you want. So it says Mac OS 10.14 plus. I'll send you the link as well. Okay. So let me click. Yeah, if I click download, it brings me down here, and it should tell you your recommended settings. So I'll paste this link in the chat as well. Okay, but if it doesn't pop up here, this is the one you'd want to use for a Mac. Okay, perfect. So that's pretty much R and Rstudio. Python's a little bit, I wouldn't say it's tricky, but it depends on the type of IDE you want, because there are different ones out there. Oh yeah, let me know if that works for you when you download R and Rstudio. So try some sample code from today's lesson, and hopefully it will work. And here's the Python link to download Python. So it does say different, Python for Windows, Python for Linux, Python for Mac. So you'd want to click, Mac, and then, you'd want to click one of these. You can click Intel. This is fine. Intel is fine. Okay, so this is for Mac. And once you've downloaded that, I'll give you some time because it does, it's probably installing, etc. In terms of your IDE, this one's an open question in the sense that you might have your own preferences. I'm gonna recommend the one that we use, like Pine Biotech, and I personally like to use. It's Visual Studio Code. So Visual Studio Code. You click that, and then it should have your options here. So Mac OS. So I'm gonna paste the link here. Okay. So yeah, let me know. Hopefully, let me know your progress with everything so far. You guys can maybe run some of this sample code. This is Python sample code. Oh, well, you might want to change the path. And I'll give you guys some R sample code. Sorry, this R code should work because it's an online link. So the R code is on the bottom. That should work. But the Python code, you have to change the path. So this is my local directory. So you'd want to change it to your own. So let me know if you have questions or if I'm going too fast. Everything okay? Purity, P, Joseph, Kisembo, Aluwade Milad, Dr. Candice, and Rebecca. How's everything? Good. Everything installed? Yeah. Oh, you're installing. All right. Okay, great. Right. Downloading. Oh, okay. Nice, nice. Yes, yes. Nice. Okay, great. If you can run the sample code in your system, that means everything is all set to go and you're free to leave. So you're done for the installation part. So good job, Joseph. I will see you next time or next session, hopefully. All right. Yeah, I personally like Visual Studio Code because I like the way they color code everything. It's just easy to understand and read. So yeah, if you can run the sample code in both your systems, then you're all set. But let me know if you have any particular questions. Okay. Okay. Yeah, let me know your progress, and I'll help you if you're stuck. Yeah, this part's the more technical part, and makes sense if it's very important though. Nice, nice. So I guess R is working for you, Rebecca. Hopefully, yeah. Take that as a well. Okay. Okay. Oh, okay. Yeah. Maybe. If you. Yeah. So I think which one, Python or R? So R, or it's fine if you say both, but R should work because it's So let me repaste the R codes. So this should work, but let me format it in the right way to let me show you how it needs to be written. So so it needs to be written like this. Okay. So you can get rid of that. That might confuse you. So this part is how it should be written. So this part, this is extra code, but this is how it should be written. So let me know if that works. And if it doesn't, still let me know. Okay. Yes. R is going to work. Awesome. Python, it might not work. And the reason for that is because you need your exact path. And the way you write your path, you need to write it in you need to write it in a way that it's backslashes. So you can copy the path name on your to your file. So let's say I go to desktop test files. So this is where all my files are located. So then you go up here. This is the path. So you copy that path and then you paste it over here. So this path needs to be put within the quotes, the single quotes. Okay. But this, all the slashes have to change orientation. So it can't be like this. This is for all your future paths as well. It needs to be the opposite orientation. Okay. It's just like that. So that's just the way it is. That's how it reads it. Okay. So you change that slash and then it should work. Okay. Take a moment to try that. Okay. So how's everyone else doing? I only Only Candice responded, so I assume everybody else is doing okay. If you finish, then you're free to leave. So, but if you're struggling, please let me know. Okay, no, that's it, Rebecca. We just wanted to make sure you have the IDEs and the programming languages installed and you can run them comfortably on your computer. So now, technically, you're already a programmer because you ran this. So congrats to you. You're you're good for that part. So you're free to leave, Rebecca. So I'll see you next session or the session afterwards. No problem. See you. So how's everyone else doing? Kusembo, Alu, Wadi, Milad, Purity. Okay. All right, awesome. Yeah, all right. So it works on your computer, Kisamba? Python? Yeah. R works? Oh, okay. I guess, yeah. Please install Python as well. And oh, not yet. Oh. Do you know how they do it, Kisamba? Do you remember the steps or? No? Oh, okay. Yeah. Well, are you a Mac user or a Windows user? Mac? Oh, okay. So check out this link. So open this link and sorry, on a second. Oh my God. And click this Mac OS 64-bit installer. So that's the Python portion. Okay. And then after you click that, you need to click, I'll send you the Visual Studio Code. So you have to install two things. So after you click that, then you have to click Mac OS Stable for Visual Studio Code. So this part on the screen. Okay. So how's everyone else doing? I only hear Kisambo and Dr. Candice, but I don't know what Purity and Olowade Miled is up to. Okay. I think maybe check if your quotes are correct and check if everything's backslash and make sure your text file is this name. So after the upmost directory, then you have a backslash and then you have the name of the file that you're trying to open. So this is the name of the file you're trying to open. This is the location to that file. And then it has to be in quotes. And it has to literally look like what's on my screen. So like every part has to literally look like this. Okay, except the path. Sorry, except the path. The path is different for your computer, but the name depending on the file you use can also be different. Okay, so this is the path to your file up to here, and then this is the actual name of the text file that you're going to use. Okay, so hopefully, how's that going? Is it working for you now, Dr. Candice, or almost on the right track? Are you still getting the same error? You're getting closer. If you feel comfortable, you can also share your screen. Okay. Yeah, that's programming. Welcome to programming. So yeah, if you feel comfortable, you can share your screen. You don't have to, but that might speed up the process. So it has to look exactly like my screen, except the path needs to be different. So everything here needs to be the same. Oh, okay, okay. Try now. I think you should be able to do that. Yeah, you should. Yeah, you should be able to share now. So yeah, perfect. Okay. Oh, okay. So you didn't download Visual Studio Code yet. Okay. So Visual Studio Code is you need to use that. Okay. Because the reason for that is, I'll tell you why. So how would I say this? So did you try downloading Visual Studio Code yet? So right now I see the IDL shell. So let me show you what I'm talking about. Okay. I'm just going to show you what I mean. Okay, so what you have is this. So right now you're here. So what you want is a more user-friendly environment. So here, this stuff doesn't work as you think it would work. Okay, over here, you might have to do one thing at a time. So this, yeah, see this stuff, some of this stuff won't work, okay? So you know what? Don't stress about this part. Let me do a simple thing. Can you write print hello? Can you write that? Does that work for you? Let me know if that works for you, because right now you don't have Visual Studio Code installed. So you didn't install this yet. So you have to click this stable down here, this blue link. So I'll give you this website and you need to install that blue link, that blue arrow down here. Because you're a Mac user, so you need to install that. That's one reason why it's not working. Another reason is, so you know what? This one, you don't need that part. This part is fine. There's some extra packages. Where's Python right here? Here. Let me see if it can work from the way you have it. As you know. No, see, it will never work that way, unfortunately, because you need to install different packages. So for that part, you know, as long as you have Visual Studio Code and Python installed on your system, that should be the main part. Beyond that, so if you want to install this, you need to write this on the bottom parts. So don't worry about this too much, but that's what you should write. And you should also write this. So this is installing the packages. But you have to have Visual Studio Code first. So you need this thing first. So the thing that you have is this one. That's what I saw. But you want Visual Studio Code. You want this thing. They look different, right? So one's more not graphically friendly. The other one's more graphically friendly. Does that make sense, Dr. Candice? So once you, so you can share your screens, right? Yes, awesome, great. Okay, now click create a new file. So there should be a, yeah, you can, I guess you can click open folder, or there's on the bottom, is there not a create new file somewhere? There's no create new file option. Okay, let me open my version and see. There should be a file on the top right, a file option, top left, sorry, top left. There should be a file option and you should click file and then new file. Or that's top right, sorry, it's top left. Is there not a file option on the top left? Oh no, I guess it's not there. That's weird. Oh no, because you're a Mac user, so you have to go up to the, like where you normally see file and all that stuff. There's a toolbar up top. Yeah, perfect. And then you select your language. Click select language. Then click Python. So type Python, sorry. Yeah, click there. Okay. And now it says, do you want to recommend? Yeah, install it. Yeah. Yeah. So this is just, it's an extension of Python into Visual Studio Code. It just helps support, it just adds more options for your your future Python coding practices. So you can go back to Untitled. So back to top all the way up to welcome, where there's welcome. Top left, welcome, Untitled, extension, Python get started. I wish I could annotate. It won't let me. So you see imagine the topmost part of your tab. Yeah, welcome. You saw welcome. You just passed it. Yeah, go to the right. You see Untitled. Yes. And now save this file just like you did. You opened a new file on the top left. Save this somewhere. Okay. And save it as a Python file. It says all files, but click all files and save it as a, yeah, Python. And name it whatever you want. Okay, perfect. It says you have selected. So click select Python interpreter and click the last one, the 3.96. There you go, that one. Perfect. And now you can copy the code that I gave you earlier. So I'll recopy it for you. Okay, here that won't work because you're missing quotes. And I'll copy the exact same code that I gave you earlier. So here's the code from earlier. I pasted it in the chat. So get rid of all the lines there and paste the code I have. Except, no, keep your path name, sorry, because you want to copy the path into the code. I put it in the chat. Okay. So yeah, go to the Zoom chat. Yeah, perfect, great. Get rid of the DF in the, yeah. So I would copy, go to import pandas and make that a new line. Start from the I for in line 2, and yeah, click there and press enter. Perfect. And now copy that path name from the backslash, or from that slash, just the path. Okay, everything in the quotes. Yeah, okay, that too. Yeah, copy that and paste it in the bottom path. Yeah, and now put a single quote in the beginning of that backslash on the bottom. Before the slash, put a single quote. Okay, now change all the slashes to a backslash. They're all forward slashes. You need to change all of them to backslashes. Okay, and now get rid of the number line and number 1 and 2. Get rid of those two lines. Line 1. No, that's line 2, 3, and 4. Yeah, perfect. Line 1 and 2. Get rid of that. You don't need that. Okay. Click that green arrow on the top right. Yeah, click that. So click it one more. Okay. Now go to the bottom where it says Canvas MacBook Pro. Yeah, over there, type PIP and then space, install, and then space, Pandas. PAN, yeah, perfect. And press enter. Oh, okay. Type pip3 instead of pip install pandas, pip3 and then space install pandas. Okay, great. Okay. So now you have to do the same thing. So pip3 install Numpy instead of pandas. So do the same thing, install Numpy. Yeah, and enter. Okay, great. Now run the code, it should work. So the top green arrow, that means you're running the code. Hurrah! Great. So you're done with both R and Python. So, due to time, I didn't think it would take so long, but I will help you guys out. Dr. Mohit will help you out in the next session on Friday to whoever needs more help with the programming aspect. And we will, we'll help you on Friday after the evening, or after the session, if you have more issues with the programming aspect. But do come earlier if you also feel like you need help, depending on how Dr. Mohit, depending on if he can handle that or balance that. And you can also meet me. I usually teach the Monday session, so you can meet me after near the end of the session to also talk about your programming issues. All right. So hopefully everyone learned how to install both, and if not, we'll talk more about it in the next session and the session after that. Okay. All right. I will see you guys next session. All right. No problem. All right. See you, Candice. All right. See you, Purity, Kasembo. Well, and use the Slack channel. Use this. Take screenshots and put it on the Slack channel. Someone will definitely get to you on the Slack channel. Okay. I'll paste the link to the Slack channel just in case you don't have it already. Okay. 1 second. Just. Could you type your emails and I'll send you a link if you don't have a. Could you type your emails so I can send you a link? Okay. Please send your emails and I'll add you to the Slack channel. Oh, okay. Candice, you're already in, but whoever else isn't in, I'll add you to the Slack channel and then I'll see you guys in a future session. All right, you're free to leave, Dr. Candice. It was nice helping you out. Oh, Kisembo there, perfect. Okay, I will add you. Okay. Sorry, just because of time constraints, I'm gonna have to head out right now. But I'll add you guys to the Slack channel, just if you aren't already there. Okay, yeah. See, some of you guys are not here already, so I'm gonna add you guys. Okay, I added Kisembo. And I'm going to add Owala D Milad. Purity, if you're there, please give me your email and I'll add you to the Slack channel. If not, you can always ask next session. Okay, so hopefully you guys got the invitation and I will see you next session, okay? All right. All right, see you guys. Okay. Bye. Bye. Do you guys get the invite? Yes? Yeah, okay. That's fine. Okay. Bye.